how you would go about defining it, I think, if you're trying to look at courage.
Right. That's exactly the kind of argument I had in my paper, actually. I think the Aristotelian, both Aristotle and Socrates bring up the idea that for courage to exist, fear also has to exist, and it's more a function of suppressing your fear. So that's kind of what I focused on, like what kind of mechanisms in the mind, in the brain, suppress fear. And yeah, it was really super interesting. And like philosophy, I've always wanted to get more into philosophy because I love having abstract discussions, and it was a really great opportunity for me because I love neuroscience too. So yeah, it was really cool.
So one of the interesting things that's come up over the years for me is I've ended up being a go-to person in the press, often after people have died doing things that are incredibly dangerous, like jumping, like base jumping, where most base jumpers eventually actually get killed. They either quit or they get killed, because you can only do it so many times before something disastrous actually tends to happen. And so I often actually get these calls about, what is it for those people who can do it? And we've actually scanned a few of them over the years. I've always wanted to actually do a larger study of it, but it's a little bit hard to pitch it, for instance, to National Institute of Mental Health, because as far as that's not a disorder. Being, showing that you're courageous isn't necessarily a disorder, even if it's incredibly risky and eventually often, has heightened rates of death connected to it. So I've never quite figured out how to pitch that one, even though I've wanted to do it on a larger basis.
Yeah, that's super interesting. Thank you. Yeah, I've never really considered, like, I guess being a daredevil, like, or like readily risking your life as an illness, but it kind of is in a sense, isn't it? Because
it removes you from the gene pool.
Right. Yeah. Do you want to give it like another minute, and then
Sure.
Hannah and Dr. Zald, do you guys get to start?
Sounds good to me.
Yeah, we could wait another minute.
Okay.
How big is the audience usually for these now that you're doing them online?
On average, we've been getting, so our first general meeting we got 30, which is a bit higher than usual. It's between 20 and 25, right? Is that accurate?
Yeah, 25. I think for our first meeting we had around like 30 or so. So
wait, we're getting a lot today actually, which is nice.
Yeah, you're 23.
I gotta say, it's a crazy start time to do these. It is not the latest talk I've given, because I twice gave talks for midnight movies. Particularly, these were shown by a movie theater in Nashville, which would show movies on, like, horror movies on Friday the 13th. And so, for instance, a year or so ago, I did a Friday the 13th movie on Friday the 13th, where my talk starts at like 11 o'clock at night so that we can start the film right at like midnight on the Friday. Those are by far have been the craziest talks to start that late, but this is up there in terms of that.
Glad we're climbing up the list of late talks we've given.
Yeah, okay. I think we could just get started then. So today we have Dr. David Zald here with us today. And Dr. David Zald is the director for the new Center for Advanced Human Brain Imaging Research at the Rutgers Brain Health Institute. He uses functional neuroimaging and neuropsychological techniques to learn more about how the brain regulates emotions. So Dr. Zald, you can take it away from here. Thank you so much for being here.
Let me see if I can share a screen here. Looks like I can, so let me blow that up. Yeah, pinwheel of death, here we go, good. Okay, I have no idea why there's this little Google thing going on here. Hopefully that will go away. So let me just sort of tell you a little bit about my background here. And I'll sort of trace my way back to a little bit about how I got to what I do these days. Hold on a second. I'm gonna make sure my screen will actually go. And what I want to actually sort of get to with this is a little bit of this sort of balance between whether I count as a neuroscientist, an affective scientist, or a cognitive scientist, and give us our argument for why studying emotions is actually super central to studying cognitive science. I am having screens that don't want to move, so give me a moment here. Let's try this again.
I think if you click on the arrow.
No, I should be okay now. So I did not plan to become a scientist. When I was in college, I was a music and film video major, as well as a Near Eastern Studies major. I wanted to essentially either do something along the lines of ethnomusicology or to write movie soundtracks. But I made a massive shift right around the time that I was finishing my undergraduate work and decided that I wanted to go into psychology and started taking an abnormal psychology class, still having no intention of doing anything in terms of neuroscience. And I started reading some books. I read a book called The Boy Who Couldn't Stop Washing, which is by a psychiatrist by the name of Judith Rappaport, which was about obsessive compulsive disorder. And I started reading about obsessive compulsive disorder. And there was a study that came out by a group at UCLA in which they found that individuals who had obsessive compulsive disorder had hypermetabolism within the ventral part of their frontal lobe or in the orbitofrontal cortex.
Doctor,
yeah.
I don't want to interrupt you. I don't think you're screen sharing.
It's not sharing. Okay,
No.
I apologize. The trials and tribulations of Zoom. So let me try to share this again. At least I'm not appearing as a cat.
Yeah, we can see it now.
So let's see if this, okay, can you see that?
Yeah, it's perfect. It's like covering the entire screen. You're
Okay,
good.
now let's see if it actually lets me advance. So this study that I was interested in, though, had been published by Schwartz et al., where they found hypermetabolism here in this area, the orbital frontal cortex, which is the same area that got damaged in the famous case of Phineas Gage, who of course had this tamping rod shot through his skull, through the middle of his frontal cortex, taking out a good chunk of that orbital and medial frontal cortex, and having this massive change in behavior that's been written about now, for over 140 years. And so I got really interested. Well, what is this brain region doing? And this was actually largely my route into studying the brain. In fact, this is actually a sample of a human specimen, just that we pulled away the bottom of the front of the temporal lobe here, taking off the temporal pole to expose this entire region just to get that sort of fine-grained anatomy of this. Now, the techniques that were available when I was in graduate school, one of the techniques that you could use to look at brain function was PET imaging, or positron emission tomography. And so, for instance, in the Schwartz study where they were looking at metabolism, they had used FDG, or F18 deoxyglucose. And what you do is you inject that into the person's bloodstream, and it will get taken up in the brain. And wherever there's more of that glucose metabolism, the more that area will shine brighter, essentially, when we pick it up with these crystal rings of a PET camera. So one way to do this is with FDG. Another way to do this is to just measure blood flow. And so you basically take O15 water, you inject it into the bloodstream, and that O15 breaks down incredibly quickly. You can only actually use it with any given injection. You can maybe get 90 seconds or so of activity, but you can then sort of have a period of time where you're just doing one thing for 90 seconds, and you could measure where in the brain you had the most blood flow, which would correspond to where in the brain was having the most activity. And you could contrast that, for instance, by looking at it between a baseline and then a stimulation sort of task. And so we started doing this. My first PET study is a study we belovedly call the FART study. The FART study came about because I knew that the orbital frontal cortex had olfactory representations. We knew this from animal studies. And we figured we might be able to trigger emotional responses very well with olfactory stimuli, since we mainly process olfaction and just as sort of like, how intense is it, how unpleasant it is, how pleasant it is. And I was trying to find some really bad odors, and it turned out at the VA Medical Center in Minneapolis, where I was working, they had one of the world's experts on intestinal gases. Now, I have no idea why anyone chooses to become a world expert on intestinal gases, but I could go to this guy's lab and we go like, can you give us some like really horrendous smells? I mean, we don't want, we don't want like people's actual farts, but what we do want is like the chemical composition. And he's like, oh yeah, we'll mix up this combination of sulfides, and he'd give us this sort of little bag of sulfides, and we could then just sort of release it in front of the person's nose while they were in the scanner. And the PET scanner's open enough that I could basically get in there and I could release it. Now, in truth, I'm getting a little bit of radioactivity exposure. I had to be monitored to make sure I wasn't getting too much. The smells were absolutely horrendous. I mean, they were so bad that we would often start giggling. There's just like, oh my God, this is just so over the top bad. But when we did this, we got, in fact, beautiful activation, both in terms of the orbital frontal cortex, but also remarkably just huge activations within the amygdala bilaterally. And so any discussion of emotions almost always comes back to at least a partial discussion of the amygdala. And this was actually one of the first really good demonstrations of amygdala activation using any form of neuroimaging. I went through a period which I referred to as the evil Dr. Zald stage, where we started looking at other sorts of stimuli that might also activate as well. So we exposed people to horrendously bitter substances like quinine. We also had a sound collage, which was this combinations of like nails scratching, dentist drill, styrofoam, metal screeching, all like on top of each other, again showing excellent engagement of the amygdala. And we could very clearly show that this was a region that was highly responsive to aversive stimuli across different modalities. The fact that we were in these studies seeing both the amygdala and the orbital frontal cortex was really interesting. It made a lot of sense because there's really strong sort of connections both between those two brain regions, but also if you look at sensory systems, all sensory systems actually project both to the orbital frontal cortex and to the amygdala, so they can both work somewhat in parallel. And then they have strong connectivity both directly as well as through the thalamus. Over time, one of the things I became interested in was moving beyond just saying, okay, these areas activate during emotional stimuli, but how does this actually interact with other systems? For instance, how do the emotionally salient stimuli impact other cognitive processes, things like decision-making or attention? And that's what I'm gonna really focus on for most of this talk. And when we think, for instance, about decision-making, often we're talking about decisions about reward, choosing rewards, hedonics, what we think is particularly pleasant, and how do those hedonics influence our choices. We're often talking about motivations, about what we're doing, what we're willing to pursue. And on the flip side, our sensitivity to loss, avoiding aversive experiences, avoiding punishments, avoiding costs. And then I'll talk a little bit about our studies related to attention. So some of the first clues, though, that this may be important, and by the way, I have a cat who seems to be attacking the carpet behind me. So if something like comes collapsing down, we'll know why. The joys of doing everything from home. So one of the things, though, that was really interesting that came up through single cell recording studies in monkeys in the very late 90s was a study by Wolfram Schultz and his student Tremblay, where they were recording from cells in the orbital frontal cortex, and they already knew there were cells in this region that responded to foods. But what was striking was those responses changed depending upon whether the food was the best food available at the time or was a less favored one. And so, for instance, if the monkey likes apple and its other alternative was a piece of lettuce, the cells would respond a lot for the apple. But now if he had a choice between, if there was a potential to get an apple or a banana, the apple was less preferred. And so now when it saw a chance for an apple, those cells actually were much less responsive, suggesting here that it was actually coding for the relative value of the reward rather than just sort of coding for a sort of constant absolute reward value. And in fact, if you started to look at this in terms of neuroimaging studies, you could do studies, for instance, where you're doing menu selection, and you'd find that the orbital frontal cortex would light up during the choice when they were making choices versus not making a choice, or making choices related to particularly high incentive foods. And you could show that these responses were not just about liking, but were often about preferences. At a later point in time, working with a graduate student by the name of Michael Treadway, we were very interested in the neurotransmitter dopamine. And of course, dopamine as a neurotransmitter is widely implicated in certain reward processes. Of course, many drugs of abuse like cocaine and amphetamine particularly manipulate this system. And we were reading some studies by a guy by the name of John Salamone, who actually gave a talk here just last week, a virtual talk. And what John was doing was studies with rats in which the rats would have a choice. They could make a decision to scale a barrier to get a higher reward, or they could not scale any barrier and they could freely get access to a lesser reward. And what John was doing then was he was manipulating the dopamine system. And so he'd often do this in a T-maze. So for instance, something like this, where the rat's in this maze and it can choose which way to go to get the higher effort, though it's going to have to expend more higher effort to get more reward, or low effort. And he'd do things like he'd give a drug like haloperidol, Haldol, a traditional antipsychotic that will essentially block D2 dopamine receptors. And what he'd find in those situations is that the rodents would choose far more of the low effort, low reward option. And they would do that instead of selecting what they normally would have done, the higher effort to get the higher reward. This is an interesting paradigm. For one thing is that the animals themselves, it's not that they don't like the food anymore or that they're less hungry. There's no evidence of that. It doesn't change their food selection if they don't have to work for it. The same amount of food if it's freely available. And it ties in very nicely to some work by a guy by the name of Kent Berridge at Michigan, who has strongly argued that dopamine is not about liking, but it's about wanting. And part of that comes from studies where they measure the hedonic responses of facial expressions in rodents, and the rodents don't change their facial expressions when you manipulate dopamine. And the idea that John put forth was that dopamine really facilitates the willingness to overcome efforts or effort costs. And so we got really interested, could we do something similar in humans? Now, it'd be hard to just like operationalize that as barriers. I mean, we could put over a wall, have people climbing over them. That didn't seem very practical in the lab. But we also knew that you could do something similar just with number of lever presses that a rodent would make to get a reward. And so we basically did this in humans. We just put together this task we called the effort task, in which the people on each trial would be given a choice, given a sort of screen in which they were shown what's the likelihood of getting a reward, how much of a reward they could get if they did the hard choice, and how much reward they could get if they got the low choice. And then the way we initially did it was always that the low choice, you'd get $1, the high effort, you would get some multiple of that. And the task would be this, and you can practice this yourself. One would be a set number of presses using your dominant hand, just using your index finger. And I want you to just go, let's just say for the sake of practice here, you're gonna do 7 taps with your dominant hand index finger. Just go ahead and do 7 taps of that. Not hard to do, you can do that pretty easily. The alternative was a bunch more taps using your pinky finger. And go ahead and do like 20 taps with your pinky finger right now. You can see, you can do it. You're fully capable of doing it, but it quickly actually is work to do it. You have to overcome some muscle fatigue to do it. And we applied this, and it actually worked really well. For instance, if we did this in patients who had major depression, they would be far less likely to choose the high effort, high reward conditions. If we gave people amphetamine, we could manipulate their willingness to do the high effort option. Interestingly, and rather surprisingly, it made no difference if you gave someone amphetamine, which causes them to release their dopamine stores. If you gave them this and you were getting high probability, where it was like, yeah, if you do it, you're gonna get the reward, that had no effect. People were always willing to do the hard effort in that situation to get the higher reward. Where it really came out differently is when it was a low probability situation, which is interesting. It's a waste of your energy typically to expend the effort if it's only a 12% chance that you're gonna get rewarded. And yet this is where you saw the biggest difference. And this may be relevant to, for instance, situations like bipolar disorder and mania, where people will do things where it's actually unlikely to work out very well for them. High risk situations, but they're willing to go after it anyway. And it's a nice example though of, we often think of, like, oh, whoever expends the most effort, that's like great, they're high achievers. But in fact, this is sort of costly to expend your effort when you're unlikely to really get much payoff for it. So these are the sorts of decision-making tasks, though, that we started to play with in order to study these processes, both within psychopathology, but also understanding the pharmacology of it. I want to flip, though, for the rest of the talk to talk about attention, because we were seeing these great activations of areas like the amygdala and orbital frontal cortex. You can ask the question, well, what's the impact of it? What's the impact of those emotional stimuli? And I'd argue that one of the big impacts is what you pay attention to. And so, for instance, if you're faced with two dogs, you can say, yeah, this one's cute and cuddly, but probably the one you should pay attention to is this guy who looks like he could chew you up. When we talk about attention, we often talk about two different types of attention. One which will be top-down modulation or goal-driven attention, or internally driven attention. For instance, when you're told to pay attention to something and you force yourself to pay attention to it, versus stimulus-driven attention or bottom-up processing, where the stimulus itself grabs your attention. Their innate properties or learned properties of that stimulus that automatically capture your attention. So how do we go about studying that? Well, one of the things we did to look at this was we sort of borrowed a tool that we knew existed from a cognitive psychology toolbox called the attentional blink. And what they do in those paradigms is they have someone having to do something like detect a target in a stream. And so in this case, it's detecting a letter in a stream of numbers. And what you find is after you detect a first target called a T1, there's a refractory period where it's much harder to detect a second target. It's like once your attention's been captured by this first one, it's not released for a while and it doesn't allow you to redirect to the next target. Right around the time we were sort of thinking about this, Adam Anderson, who was a doctoral student with Liz Phelps at NYU at the time, published a paper in which they used a paradigm in which they did this sort of attentional blink, and they had people looking at a stream of words. And what they found is, and they just had to detect the words that were of different color, but they found if the emotional words, if the second target was an emotional word, it often broke through. And it would break through that refractory period and still be able to capture attention. So working with a student by the name of Steve Most, we started sort of playing with this idea and say, well, what happens if you've got an emotional stimuli to begin with? Will those stimuli capture attention so much that you couldn't redirect your attention? And for instance, we wondered, for instance, would emotional stimuli like threat stimuli do this? Would potentially sexual stimuli do this as well? And so we just sort of developed this paradigm called the emotional attentional blink, in which you had to view a stream of pictures that are going by rapidly. And your job was just to detect a target that would be a rotated picture. But some point before that picture, we would drop in an emotional stimulus. And the question was, would an emotional stimulus here capture attention and block perception of something, of a target in this scenario? I'll note, this is actually Stephen Most, the graduate student who did this work. He's now a professor at the University of New South Wales in Australia, where I guess they are a little bit rougher, even in comparison to New Jersey, perhaps. So might give you an example trial. We'll see if this glitches. Hopefully it will come across despite it being on Zoom. So your job is just to look for a rotated picture. It will be the picture that's rotated will always be a picture where there is a clear sky on one side of it. It could be a building or it could be a landscape. So I'm going to give you one example trial here. Anyone see a rotated target in there? I'm going to give you another, for those of you who didn't, I'm going to give you a second try on that one. So take a look this time. So I hope many of you could see there was a rotated tree in there for one of these. So now I'm going to do one where we're going to have at least one stimulus in there, which is not a neutral stimulus. And so try it this time. Ready? Go. Okay. Did anyone see a rotated target on that trial? Anyone see a gnarled hand on that trial? So to show you that there in fact was a rotated target that happened just after that gnarly hand, this was actually what you missed in there. There was this rotated target. When we did this study, what we found was that we had, if we gave someone a scrambled picture in there as the distractor or a neutral picture, it really did not make a difference if it was just a couple lags before the target. But when we introduced a negative threat picture or gory picture, it had a huge impact and people had a much harder time being able to detect the targets. It wasn't just a surprise-induced blink. There is a phenomena called a surprise-induced blink, where the first time that you see something that was like, that was unexpected, it will grab attention, but it will only be able to do that about twice. Whereas our stimuli, we could do this for dozens and dozens of trials, and the gory stimuli would still have the same effect. It clearly was not just aversive stimuli. The next study we did, we took a bunch of college-age heterosexual males. We introduced either as the distractor image, it could be a clothed male, it could be a clothed female, or it could be a nude female. And we saw that the guys did horrendously at it if we introduced a nude picture of a nude woman. In that original study, we actually did a follow-up where we paid them for good performance. We said, okay, maybe you just want to look at the naked women. We'll give you money to do it well. The money made no difference. And you can think of this as sort of a true type of bottom-up attention in these nude pictures. We actually, in those studies, those first studies, we did this in a way where chance would have been 50%. We ended up changing the way we calculated it such that later where there couldn't be guessing. And we found that the performance was just atrocious, that they could only actually get like less than 20% of the targets correct. This is not limited to men. We do lots of our studies where we'll do, for instance, nude couples. It's clear women are responsive to this as well. Though I will tell you, the college-age males are truly, truly deficient when it comes to this. They truly struggle with this. This study got probably more attention than almost any study I ever did. Because people were able to put in headlines like "gore and porn make you go blind." It even got tagged by the New York Times. They used to have a Year in Ideas episode, and they included this paradigm in there. Mainly because of the press it got, because of this idea that looking at porn makes you go blind. People just loved that old wives' tale idea. The part of the reason this may work is that there's actually really robust connections between emotional limbic brain regions and visual sensory processing areas. So for instance, the amygdala receives projections from all stages of from late stages of the visual processing stream, but then projects back to even the very earliest stages of visual cortex. And for instance, if you show people pictures that are aversive or erotic relative to neutral pictures, you'll get massive activation of those sensory cortices. It's an incredibly robust phenomena, and you can do all sorts of matching. You can do this relative to scrambled pictures. They can be matched for color, all sorts of things, and you will get this robust engagement. Sensory systems are incredibly well tuned to emotionally valent stimuli, to stimuli with value. Another sort of paradigm that you can use to get at this sort of same thing, to study like how much people can actually control this, it pulls off on the Stroop task. So you all know the Stroop task, in which people normally, the way it normally works is you have to name the color of a font, but you're doing that in words which are named the colors. But you can do something similar for words of any kind. So if you just wanted to sort of like go through this list here of neutral words, ignoring what the words say, and just for yourself, see how quickly you can read through just naming the color of the font. So just going, red, blue, green, red, blue, red, green, red, blue, green, red, blue, green. Now I want you to try it on this next screen. So how many of you felt that you had to fight a little bit harder to focus on the color and ignore what the words were saying? And it's these sorts of paradigms that we've used to try to get at individual differences in people's ability to balance goal-driven attention and this sort of bottom-up sort of attention. What I hope, that is sort of coming through on this, it would be an argument that these cognitive processes are often really highly influenced by value of stimuli, be that positive value, motivational significance, that could be negative or aversive value as well. And that heavily impacts both decision-making and attention. And as such, we really need to be including those affective processes when we're thinking about cognition. We shouldn't be divorcing them and treating these as separate areas of study, that these are in fact inherently influencing our cognitive processes, and to think otherwise would be highly problematic. So I just then, what I want to do just with my remaining time here is tell you a little bit about the center we're opening up, and what that's going to look like. And then I'm open to any questions. So the Center for Advanced Human Brain Imaging Research is going to open up on the Bush campus. We're hoping to have it open in June of this year. Construction has just started. There's a building back here behind the Richardson Apartments, and this is the United Behavioral Healthcare, and it's just tucked in here. You'd have no reason to ever know this building existed, but it's called the Staged Research Building, and we've taken over 1/4 of this building to put in a new neuroimaging center. And we're basically going to have both offices where you've got a scanner, a control room for that scanner, a mock scanner room, which we hope to eventually put a second scanner in. We've purchased actually a Siemens 3T Prisma magnet, which is sort of a state-of-the-art magnet with their state-of-the-art head coils, which will be delivered probably the first week in April is what we're hoping for this year. This will allow us to do really high resolution structural imaging. It will allow us to do fMRI at high resolution down to about 1 millimeter thick voxels if we were to choose to. It allows us to do detailed mapping of white matter in the brain. It also allows us to do a technique called magnetic resonance spectroscopy, where we can measure, for instance, neurotransmitter levels of certain neurotransmitters. And we can measure, for instance, blood flow as well using these techniques. And so the idea is to really to be able to build a research center allowing state-of-the-art cognitive neuroscience research to be done here on campus. And I was hired basically to start this center, and really excited to be able to move forward on it. It's taken a while to get this thing going. The pandemic certainly has not helped us in this regard, but we hope to have that we will have this open this June, and there will be opportunities for students who are interested to get involved in research here, both in terms of if they want to be subjects and get their brain scanned so they can get a picture of their brain, as well as though if they wanted to be involved in neuroimaging research, those opportunities will become available. So I'm really excited that we're going to be able to do this. And that's all I wanted to talk about. I'm happy to answer questions, whether they're broad questions about cognitive science as a career in neuroscience, or whether they're narrower about the sort of topics that I chose to speak about today. Happy to answer them. And I will stop sharing my screen here.
So I have a quick question.
Yeah.
The center that the department had for
Yeah.
is that going to be focusing? I assume it's going to be focusing mostly on your research and your projects first, right? And then you'll be.
I would not.
How does it work?
I would not put it that way. I mean, the initial very short ramp up period will just be making sure we can optimize sequences, but the doors will be open for researchers. Now, the critical thing though is neuroimaging is not cheap, and this is the challenge, because these scanners are expensive. I mean, this was the scanner itself cost us over $2 million. The renovations and putting the shielding in the room, there's end up being another $1 million or so investment there. And so the economics are, over time, that has to be paid for through grant money and people getting grants. So early on, a lot of the focus is going to actually be, we've started a pilot grant pilot grant system that researchers at Rutgers are going to be able to apply for funds to basically pay for scan hours, so they can start collecting data and then use that to apply for grants. And so it's very much not meant to be my research first. Really, my goal is to be facilitating that for researchers on the campus.
Okay, thank you.
I think I have a question too.
Yeah.
So first, thanks for the talk. The demonstration is really engaging. And did you find any differences between positive and negative stimuli?
So it's interesting. It turns out that for sort of more typical positive stimuli, for instance, if I give you pictures of food when you're hungry, I'll get a little bit of an effect. By far, the largest effects we've ever seen are for erotica. They are just sex grabs attention to an incredible degree. And it's striking because one of our studies that we published a couple years ago with one of my collaborators was of patients with PTSD. And we took, these were military vets who had been combat exposed, who had post-traumatic stress disorder. And we showed that we used our emotional attentional blink paradigm, and the distractors were sort of combat related pictures. They clearly were highly captured by those relative to controls. Even in those subjects though, even in those PTSD individuals, erotica caused a greater emotional attentional blink than the aversive stimuli. We have just simply never found stimuli that draw attention on average more than that. And I don't know why that is. From a sort of evolutionary perspective, it doesn't make sense, because you'd say, look, what's more, if I show you a threat, I've got someone pointing a gun at you, or I've got a naked person. Yeah, maybe I get lucky and I get to have sex with that naked person. But really, what should be my bigger concern right now is the guy with the gun. Because there ain't gonna be no chance for whoopee if this guy shoots you. And yet the erotic stimuli were even more powerful.
Thank you. That's really cool.
One of the things I will note with that is those stimuli tend to be very easy to pick out. They have large swaths of skin tone in them. But other than that, I don't have a great explanation of why that is. Now, an interesting question that we haven't pursued that I'd love to actually see, if we manipulated a system like the dopamine system, could we actually push that around? For instance, if we gave someone amphetamine, would that make them even more reactive? And we've never done those sorts of studies. I'd be curious to do them.
Hi. Okay, I think you can hear me, I hope. Okay. So my first question is why you first got into neural imaging. Like what about that was exciting, and why you decided to study psychopathology, especially concerning dopamine levels, I guess. And secondly, what sort of implications do you think your studies have in like the real world, I guess? And third, last question, what studies do you hope to do in the future with the new center coming?
Yeah, so let me sort of work my way back. There are a number of us researchers, I find, in my generation and the generation just at sort of a little bit younger than me, where these techniques were coming online. And the ability, but prior to that, like when I started grad school, I worked with a guy by the name of Richard Depew. And Richard, basically his idea was you should read, if you wanted to understand the brain, you had to spend your time reading animal studies, but then you would know that most of those studies you couldn't actually do in humans. And so you may try to make inferences from them, but there's going to be limits on what you could do. And as the neuroimaging started to become available, it's like, wait, we can actually sort of get a sense, we can sort of peer inside people's brains and see, at least in a crude level, not a single cell level, that the level of regions, what areas become activated, how those different areas are connected functionally, what their interplay is, and whether we could look at individual differences that would be informative for understanding the neural substrates of both individual differences and like personality. But as someone who was trained in clinical psychology, highly interested in whether we could see correlates of different sorts of psychiatric illness. Now, prior to that, I was also, my training was also heavily, if you were someone who was in clinical psychology and you wanted to understand the brain, the route to go was neuropsychology, where you'd study patients with brain lesions. I still study certain populations. I remain fascinated by patients like the Phineas Gages of the world who have ventral frontal lesions, who have changes in their personality following a brain lesion. To me, that remains fascinating. I've done a number of studies with patients with amygdala lesions. Usually these are epilepsy patients where they intentionally, the doctor goes in and removes the amygdala and part of their hippocampus, because that's the focus of where their seizures come from. And it's better for them to lose those areas of the brain than to have those areas constantly generating seizures. And so we've been really interested, well, what does that do to their emotional processing? And so I've always, the ability to start to look and ask those questions in a way beyond just saying, okay, this person's got this lesion, was really a driving force for me. In terms of psychopathology, that was always really core to sort of my mission as a scientist. But I think the thing that was sort of, at least I didn't necessarily expect as I sort of started my career, was actually the extent to which I paid attention to cognitive science. And I will say, as someone who got a clinical PhD focused on neuropsychology, you'd say, well, you do, if a neuropsychologist, they study patients with dementia and they give all sorts of cognitive tests. That's true, but cognitive psychology has had this whole other group of techniques for looking at and asking about cognitive processes. And I actually got very little of that in my graduate training. I'd say I still didn't get that much of it even in my postdoctoral training doing neuroimaging. I got a lot of it actually from my psychologist peers who were cognitive psychologists. And it became more and more apparent to me that I needed that. And it took me a while to get there, but like early on, I even wrote a paper which was about sort of trying to understand the orbitofrontal cortex, and you talk about its projections to the parts of the basal ganglia and the caudate nucleus, and people talk about, well, that's the caudate is the cognitive division of the striatum, the putamen's the motor division, and the caudate's the cognitive division, the nucleus accumbens or ventral striatum's more the motivational part of it, and sometimes referred to as the limbic part of it. You go, well, wait a second, what do they mean when they say it's the cognitive division? Saying something's cognitive is not actually that informative. And I, early on as a graduate student, had written a paper, and the reviewer had, said, well, that's not all that informative. And I was like, wow, they're right. That's like useless, just cognitive. What does that mean? And so to me, it's been a 30-some year realization of that all these parts fit together, and trying to understand the integrative whole, and then saying, well, how does that help us in explaining psychopathology, where, it's not all that useful to be able to think about psychopathology and go, oh yeah, it's a serotonin deficit. Well, what would that look like? What would that mean? How do you mechanistically translate between serotonin and the symptoms of a disorder like depression? How do you actually go from, okay, this area is hyperactive. Well, what would that hyperactivity mean functionally that would then translate into symptoms? And so to me, that's been the driving sort of component. Now, your last question is the one about sort of studies that I myself really will be engaging in the most. And one of my problems as a scientist is my interests are quite broad. And that means I tend to jump into a number of different domains simultaneously. But one of the things I'm particularly interested in right now is there's some techniques that have been used where we can, by measuring sort of a typical response across the brain to a set of stimuli, we can predict in someone else's, by their brain response, to what extent that person is having a response that shows a standard response, say, to a negative emotional stimuli or a positive emotional stimulus. And what I'm really curious about is how that changes in certain patients with certain brain lesions. For instance, in ventral frontal cortex or amygdala. That's one of the domains I'm very interested in. The other is really somewhat driven by some of our past work with drugs of abuse and drugs like amphetamine and modulations of the dopamine system, and asking questions about how, if we shift or modulate the dopamine system, what impact does that have on the larger networks of brain functions while the person is engaged in tasks related to reward. And those are two of the avenues I particularly am eager to pursue. Great question.
So we have a question in the chat.
Yes.
Nafi asks, what could removing those portions of the brain, like the amygdala, do to a person without seizures?
Yeah, so it's a great question. And obviously you would never want to remove it unnecessarily. There is a fascinating disorder called Urbach-Vita disease, where you get progressive calcification of the amygdala bilaterally. It's really one of the only situations where you'll get both amygdala being disrupted. And those individuals are interesting. They seem to have a lot of problems detecting fear and understanding other people's emotions. They interestingly seem to struggle. They seem to be very trusting of people, like they don't detect that other people might be out to get them, and so they're very, very trusting. They also often show some deficits in fear learning, where they don't learn to connect, sort of the pairing of a negative consequence with the situation. But one of the things that we've made some arguments about is that there's a lot of actual redundancy in the brain. And a lot of times where you really see the biggest effects is if you get a combination of lesions. It's really my belief that it's when you get a combination of, for instance, both the amygdala and the insula, or both the amygdala and orbital ventral frontal brain regions, that you tend to get the biggest impacts. What's really interesting with those ventral frontal cases is you often get what we can call pseudo psychopathy. Basically, these guys can look sort of like psychopaths. They start to become somewhat fearless. They no longer have much empathy for other people. They don't seem as mean or cruel as you can sometimes see with a traditional psychopath, but the sort of fearlessness, lack of empathy, lack of concern, high risk taking is really quite striking. You'll also see this in patients with a type of dementia called frontotemporal dementia, or the behavioral variant of frontotemporal dementia. And what happens is that ventral frontal lobe starts to atrophy at a really rapid rate. And oftentimes the first thing you see is their personality changes. They start getting into trouble. They start taking risks. They stop being as responsible. They may start engaging in gambling behavior. There are cases where they start showing, for instance, pedophilia, things that they would have normally suppressed, which get released. And so they're really interesting cases. Those are much more from the ventral frontal than the amygdala, whereas the amygdala is actually a little bit more subtle in terms of its effects. Those ventral frontal ones are really striking. It's actually one of the things that really got me interested in neuropsychology in the first place, were some of these cases, which I thought were fascinating.
I sorry, I have another question. This might be a bit of an amateur question. I was reading a book about how it was like published in the 80s, but it was talking about how we don't know all of like the brain receptors like serotonin and dopamine and norepinephrine. I can say that word, but we don't know all those receptors. And I'm wondering if you find more receptors by using neural imaging. I have no idea how people find those. So yeah.
So very little would come about in terms of discovering new receptors by neuroimaging. What neuroimaging does allow us, though, to do is do much more in terms of quantifying those receptor levels. So for instance, less so with MRI, but more so with PET imaging, we can take a chemical and we can tag it with something like carbon 11 or F18. And so I've worked a lot with a drug called falopride and another called raclopride. Both of these are chemicals that bind with D2 dopamine receptors. And so what we can do is we can label falopride or raclopride with F18. With raclopride, you usually do C11. With falopride, you do F18. It's just which one works better. And then we basically inject them into people's arm. The neurotransmitter goes wherever that neurotransmitter normally would go and binds. And then we can calculate for different individuals who has higher levels of dopamine D2 receptors in given regions of the brain, and ask questions about that. And so, for instance, some of our studies have looked at impulsivity and individual differences in impulsivity based on the dopamine system, or novelty seeking, novelty responsiveness. I've got a study that I hope to finish writing up, where we were looking at dopamine measures, in particular a measure of the dopamine transporter, and using a test of attention, which is a test called the test of vigilance and attention. And this is a measure that's often given to individuals with ADHD as a probe of the severity of their ADHD and their attentional problems. And we see some really interesting correlations between those dopamine transporter markers and attentional performance, which is interesting given that the main way we go about treating ADHD is by giving drugs that block the dopamine transporter and alter those uptake kinetics. And so it's less been about discovery and more about, okay, we know those receptors or those sites are there. How can we quantify them in psychopathology, in normal range, as well as in processes like aging? One of the things that I guarantee you, without a doubt, because I'm 30 years older than most of you here tonight, my D2 dopamine receptors are lower than your D2 dopamine receptors. They're going down. You've already hit your peak of those receptors, and they're going to steadily decline for you for the rest of your life. What does that mean? There are certain things you can say, well, young people, there's certain things they can do faster than older folks. But the level of difference is enough. The changes are dramatic enough that you go, wow, if I saw that in a 20-year-old, I'd probably be like, well, that's like a sign of pathology. And yet, like I'm a normal, healthy 50-year-old, and though I guarantee you those levels have declined dramatically, what does that mean? Now, eventually, of course, some of those declines in the dopamine system are what leads to Parkinson's. The biggest risk factor for Parkinson's is simply how old you are. The older you get, the more vulnerable you are because your dopamine neurons are dying, essentially. which kind of sucks when it comes right down to it. But sort of trying to quantify what that looks like. What is the level that you have to hit before those problems start to actually manifest? And luckily for us, they go down, the system compensates as it's declining, it's declining, and the system just compensating. And you only start getting those Parkinson's symptoms once it gets too low that the system can't compensate for it anymore. But those sorts of measures we never could have done in the past, because the only way you would have been able to look at that would be once someone's died. Let's say they got hit by a car, I could autopsy the brain, I could measure number of dopamine neurons in that situation. But the neuroimaging allows us to do this in living healthy individuals in a low risk manner.
Thank you, Dr. Zald. Does anyone have some last questions they want to wrap up the night with? Those are all really good.
I was
Thank you.
just
What's up, Cynthia?
I was just curious about like how I know you said that you started out like studying like music and like Middle Eastern studies or whatever, like how you like made the transition from something like that and then like learning all of, like the very like technical stuff that has to go with science.
Yeah. So I will tell you, there's one aspect I would consider myself to have been very lucky in, which was I was lucky in that I was, at the time, I was in state in Michigan. I had done my undergrad at University of Michigan, and it was still relatively cheap to attend. My parents were willing to support me. Basically, I finished my undergraduate degree and immediately started taking classes because I really did not have the psychology and the neuroscience background to do what I wanted to do. And I just went back sort of to school full time to get and started working in a research lab to get myself in a position to get accepted to grad school. Frankly, I was very lucky in many respects. I got into a very good psychology program at the University of Minnesota. Frankly, looking back, I probably wouldn't have accepted me as a student. I would have been like, who is this guy? He doesn't have the background. But I was committed, I was passionate, and I was willing to put in the hours to really learn what I needed to learn. And luckily had a program that was like, yeah, okay, this guy seems like he knows what he wants to do. Even then, I'd say it was a winding path for me initially till I sort of started doing the neuroimaging, and that's when everything started to really coalesce for me. But I think there's a lot to be said to being willing to, I had the opportunity to take that extra year to get myself into a position to apply for grad school. And then let's just say, I've at times not had the best work-life balance. If you're going to be broad and try to cut across different disciplines, you've got to really want to do it. And I always, once I sort of had the thing about what I wanted to study, the passion was there. And in many ways, for me, like, being a professor, going into academia is like the greatest treat, because it's like, I gotta go, well, what is it that I want to study? What is it that I really want to learn about? And then as long as I can figure out ways to both do the studies and get grants to do those studies, I can convince other people that this is worthwhile. I get to do that. I don't have someone saying, oh, this is what you've got to study. Rather, it's like, well, this would be really interesting to understand this. Let me start reading about this to see if I can understand this. And then what are the questions that we need to ask about it? And there are these sort of chains that happen because like once you do one study, it almost always brings up questions for like the next thing. Well, we saw this. Well, what about this? Well, what about this? And before you know it, you know, it's like that you're a decade in a research program. And, you know, my wife, you know, I'm old enough to be starting to talk about, you know, like when I might retire. And it's like, I'm gonna keep doing this for quite a while until I feel like I can't do it well, because every day I'm excited to be able to ask questions. Sort of like it's a privilege. It's a privilege to be able to spend your life studying things that you find interesting and training the next generation of scientists who can do that themselves. I mean, I can't think of well, the job that I would have probably preferred to have would have been to, you know, make my living as a rock star or something like that. But given that that happens to very, 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 very few people are successful at this, this is a very good second option in my book. I mean, if, you know, if I could have played in Bruce Springsteen's band or something, I would have been equally happy, I'm sure, but that's not going to happen.
All right, well, thank you so much for coming, Dr. Zald. Really appreciate all your insights. Presentation was amazing. I expect nothing less, I'm sure, for many of us. Before you guys go, please make sure to check out our link tree. So last meeting we had we introduced our mentoring program for those who are interested in neuroscience. So if anything here caught your eye or might have interested you, you can certainly learn more. As Dr. Zald proved himself, it's never too late. So yeah. Also check out Dr. Zald's SoundCloud. He has some really cool stuff on there. Stuff he makes with his instruments, which you can't see because of his virtual background. But
I'm not sure when I got the virtual background again. Let me just say, if people are particularly interested in getting involved in neuroimaging research, feel free to shoot me an email. Happy to talk to you more.
Could you just send your email in the chat really quick?
Sure.
Or you can just tell me and I can type it up if you'd like.
Just david.zald@rutgers.edu. Nice and simple.
David.zald@rutgers.edu. Well, there you guys go. Yeah, for any new people, sign up to the newsletter so you can keep up to date. And is there anything else I have to say? I don't think so.
Oh wait, can I bring up the Google form for the swag and stuff?
Yeah.
Okay. Okay. Sorry. Hello. So basically, we're working on getting merchandise and swag to you guys all this semester. So if you guys could all fill out this Google form about the certain products we're gonna have in our store, it'd be greatly appreciated. Thank you. Okay, I sent it in the chat.
I want to know what swag you guys have.
We don't have, it's not available yet, but we could definitely shoot you an email if you would like to have some swag. But yeah, we're looking into like sweaters, socks, or like masks, especially now since we gotta, yeah. Oh, we're also looking at tumblers. Tumblers are really fun. So yeah. Someone just shot me a message about bucket hats. Bucket hats are also really fun.
I would definitely game for some swag.
That's good to hear.
Thank you guys for having me.
All right, thank you so much for coming,
Thank you
especially
so
so late.
much.
Really appreciate it. And
My pleasure.
thanks to all of you for coming so late. It's not easy on a Wednesday night, but really appreciate it.
Bye-bye.
Thank you so much.
All right, bye, Dr. Zald.
you.
Thank you.
Bye.
Bye-bye.